This conference will now be recorded.
Hi, this is Joe Gallo from ANCAN, and welcome to another one of our Wednesday night drop-in sessions. We'll get into the details of that in a moment, but I'd like to just get over a couple administrative things. First off, I would appreciate if everyone would maintain themselves on mute so that we don't pick up background noise or anything to distract our presenter. If you will have any questions along the way, please put them in the chat box. The moderators will pick up the questions and present them to our guest tonight. And of course, the standard disclaimer that the information and opinions that are expressed tonight in this presentation, that includes anybody who's a participant, including our presenter, are not recommendations from ANCAN, and they're not intended for medical treatment or any other course of action. We strongly suggest that you consult your doctor with any particular situation that needs attention. And with that, I will turn it over to Howard Walensky, who will introduce our guest.
Thanks, Joe, and thanks everybody for coming. Our speaker tonight is Professor Aurora Esquela Kerscher, who's an associate professor of microbiology and molecular cell biology, and is a prostate cancer researcher at East Virginia Medical School in Norfolk, Virginia. Her work involves translating her findings into effective diagnostic and therapeutic tools for aggressive forms of prostate cancer. So I'm glad that we have sort of a mixed group tonight of men with going all the way from low risk to advanced prostate cancer. Now, she teaches a class at the medical school. There's a club of medical students who are interested in scientific research. So she lectures to them on how to read a research paper. And so she's taking that idea and has prepared an original program for us tonight entitled A Layperson's Guide to Reading Medical Research. So with that, I'll turn it over to Aurora.
Thank you. Let me just get this on the screen. How's that? Okay.
Perfect.
Perfect. Well, thank you everyone for inviting me here today. And it's just nice to be, in an audience of people who understand the field. And like you were saying before we started, from all different perspectives. So, I really come into prostate cancer in this field as a researcher, and so it's nice to actually talk to patients who are living with this disease. So let's see, here we go. So I thought I would just talk just for a couple minutes, just introduce myself and my background so you understand where I'm coming in, and then how I evaluate different research topics and how I read research papers, and then hopefully, some of the ways how I kind of attack and try to understand different papers, and reading scientific research can be, quite obscure. Hopefully, some of these tips will be helpful to you too. So, just a disclaimer, I'm not a clinician. My training is in developmental biology. And then more recently, I have transitioned from developmental biology to thinking about cancer biology. And then as my career has progressed, thinking more translationally in the diagnostics and therapeutics. But I really did get my start as a developmental biologist and using really interesting model systems. You can learn a lot about cancer pathways just using really simple models. And so I've been working with slime molds when I was at WashU in St. Louis as an undergrad, and then I got my master's in biotechnology and my PhD at Johns Hopkins, and thought a lot about organogenesis, mainly liver and kidney development using mouse models, and then transitioning to Yale for my postdoc. And this is really where I started thinking a lot about developmental pathways, and a lot of these pathways go awry in cancer, and using both simple nematode worm models, mouse models, and then at EVMS really transitioning into the human system. So at Yale, I was thinking more about developmental factors that were important in lung cancer, and then at EVMS thinking about interesting factors, actually non-coding factors in prostate cancer. There we go. So I'm in Eastern Virginia Medical School, so I'm in Norfolk, Virginia, and I'm actually sitting right here in my office right now. It's a nice quiet place this evening. But if anyone's ever around, I'll be happy to show you my lab, but just virtually you can get a kind of a view of what goes on in a research lab over here. And at Eastern Virginia Medical School, we do have a very vibrant, it's a small medical school, but a vibrant cancer biology, immunology, cardiovascular, diabetes research programs, and also historically reproductive research. And actually, the Jones Institute on campus was the place where the first in vitro fertilization was done in the United States. So we have a nice history with reproductive research as well. And then I just thought for a second, I just kind of give you an insight of what I'm working on in my own research lab, and again, how I approach my own literature searches and getting through all of the scientific literature to help advance my own program. So for me, we think a lot about non-coding RNAs. These don't produce proteins, and then think about how we can develop these really interesting factors as diagnostics and therapeutics. And at EVMS, we actually have one of the largest serological biorepositories in the nation. It's been very pivotal in building my own research program and identifying really interesting non-coding RNAs that are closely associated with cancer. And in our community, prostate cancer is a huge problem. I mean, obviously, it's a second leading cause of cancer deaths. Virginia actually has the 8th highest mortality in the nation, and where our institute resides in the Hampton Roads area, we actually have one of the highest mortality rates in the nation. So this has really impacted the community, and we've really devoted a lot of our research trying to understand how to try to develop our own research with these microRNAs to help in screening, trying to differentiate in patients, not just cancer, non-cancer, but again, we're finding some of our factors that are really good at differentiating between low-grade and high-grade cancers. So we're thinking about fluid-based diagnostic screens. And then we also have, some of these factors also have very functional roles in promoting prostate cancer. And so we have some interesting non-coding factors that will increase invasion, increase tumor load in some of these mouse models. And right now we're teaming up with some great researchers, both in Ohio and Connecticut and in Boston, and testing some of these novel reagents to block some of our factors we're thinking about, and hopefully to block aggressive tumor progression and castration disease. So that's sort of what we've been thinking a lot about. So yeah, so for today, I'm really happy that people came and are interested in trying to dive deep into the research literature. So hopefully this indeed will be a guide to help you. So feel free to ask any questions as I go along. I mean, obviously the goal is to learn more about prostate cancer research, but this could be for a lot of different reasons, right? You could want to understand the latest discoveries, both basic and translational. And as we were discussing before, to educate, to self-advocate. So you really need to, both understand the newest discoveries and what clinical trials might be out there, and then what type of basic or translational research really were the foundation for some of these trials. And so, I mean, the challenge then is not really like how to read a research paper, but the primary challenge is how to find a good research paper that really will help in whatever you're interested in learning more about. And to do that, you really obviously need to understand the field and to understand, the impact of some of these new techniques or screens, and if they'll really be useful down the line. So honestly, to get the big picture, there's a lot of resources out there, and I just listed a few. And then again, a lot of the primary research literature as well. So obviously the first challenge is finding the right paper. For me, like we have very specific questions that we're asking the lab or looking for a new technique to bring into the lab. So honestly, one of the fastest and easiest ways to kind of sift through all of the noise, I mean, there's so many scientific journals right now, and the papers are coming in, fast and furiously, is just to go and do a Google search. And, the trick is to find the right set of keywords to kind of flag some of the papers that you might want to skim through and see if you want to read the whole thing. Google Scholar is a great place to start if you want to find a research paper. Also, PubMed, which is run by the NIH. For us, if we can't find a paper, I mean, one of the concerns is that there might be a great research paper that you want to read, but it's not free. It's not free access. So the question that was posed to me is, how are you supposed to get access to those? And so for me, being from a small medical school, it's just not affordable for the school to subscribe to all of the journals. I mean, that's just thousands and thousands of dollars. So Google Scholar, if you have a title of a paper that you want to read, you can do the title of the paper and then just put the term PDF, and sometimes you can find free access to a paper that you're looking for. PubMed, usually if the investigators were NIH funded, then their papers will also be in access through PubMed. And then for me at EVMS, in our medical school library, if there's a paper that I can't get access to, then I can request it, but of course this costs money. So it's tricky. You can potentially ask a clinician or someone who has access to the libraries in a large institution. And then, if you have tons of potential papers that you might want to read, then, how do you choose from the pile? I mean, one consideration would be the impact factor of that journal, which, the impact factors are published every year. And an impact factor is a measure of the yearly mean numbers of citations of the articles published within the last 2 years. So this means that if it's a great pivotal paper, maybe it's, described a new screening tool or a new therapy. Obviously, if it's amazing and innovative, then a lot of other papers will build upon that work and they will cite that primary paper. And so that will increase the impact factor of that paper and of that journal, of the journal itself. So that's a way to figure out, if you find a paper in, say, New England Journal of Medicine or Nature, it's probably been a very well thought out and a pivotal paper in its field. And then the second challenge would be then, obviously, which I'm going to talk about, is understanding the paper. So obviously, science literature isn't beautiful literature. It can be really hard to get through. It's like reading a different language. Even a scientist like me, when I'm collaborating with a bioengineer and I have to read up on biosensors or something like that, I mean, even though we're both scientists, we don't talk the same language. So it's very hard to get through papers even outside of one science field to another. So, like, why are you reading the science paper to begin with? I mean, obviously there's a lot of different reasons why you want to pick up a journal, and that can sometimes dictate how you're going to go through and read the paper. So again, it could be for educating or self-advocating for medical care, keeping up with the new scientific advances. Maybe you're just really interested. It's a cool hobby for some of these new fields. Maybe you need to present a paper or write a review about the field, learn a new scientific method for a lab. So any of these reasons will really dictate how you're going to read the paper. And so this kind of, outlines, like, what are you looking for? Are you looking for an ideal for research? Are you looking up a diagnosis, a therapy? Are you looking up a topic overview or a new update of the field? This would kind of dictate where you would start looking for that information. For research, I'm looking a lot in original articles. I don't necessarily read clinical trials that much since I'm a developmental genetic pathway kind of researcher. So I'm usually in the primary research arena. If you're looking for a topic overview, which is great if you're getting into a new field or trying to learn a new therapy, then reviews are really great. Maybe reviews of big meetings will be published as well. So this will all dictate exactly where you would also look for that type of information. And so this is kind of just a flow chart, like you don't want to read a paper and really to be committed will take hours to get through sometimes because it's so dense. So there's, you just need a system to quickly skim through and figure out whether it's a waste of your time or not. So this might seem like common sense, but it really saves time, especially if you're writing a review or trying to write a grant or just trying to get caught up quickly on a different topic. The title can be extremely illuminating on whether or not you even want to continue reading another word. All papers have an abstract, so you can skim through there. But then you're kind of like, even going through that, I mean, is the paper well written? Does it have clear objectives, a hypothesis, clear data or conclusions? If this isn't something that's relevant to you, then just, put it aside and go to the next article. Because again, once you really commit, it does take some time. And so for today, our case study is a really interesting and late-breaking paper that Howard recommended to me, and I thought it's a nice paper that's easy to read. There's some data in there that kind of is instructive for how to critically read a paper, and obviously is relevant to us. The title is Effects of Exercise on Cardiorespiratory Fitness and Biochemical Progression in Men with Localized Prostate Cancer Under Active Surveillance and a Randomized Clinical Trial. So this is actually a great paper title because just the title has given you a ton of information and a decision of whether or not you want to now invest a couple more brain cells and think about what this paper is going to say. The good news for academic research papers is that they all have a specific format or template. And so this is sort of the anatomy of a research paper. And so from journal to journal, it might vary a little bit, but research papers, again, it's not beautiful literature. There is kind of a cookie cutter kind of standard format that scientists are used to seeing, and that helps us when we're trying to skim through too. So I'll give you some tricks. So this is the anatomy of a typical research paper. So obviously the title, the abstract, which is usually a paragraph, which gives you an entire like synopsis of the paper down from the intro to the conclusions. Usually there's some keywords for the paper. So for example, for this paper itself, keywords would be like active surveillance, cardiorespiratory, exercise. These are keywords that you would also be helpful when popping up if you're doing a Google search looking for clinical trials with active surveillance and exercise. This would pop up. Introduction, materials and methods, a results section. There's a discussion section, references, acknowledgements, supplemental data. So these are the very typical specific subsections of a research paper. And so for our case study, I'll just kind of go through all of the sections and just tell you, how to get through them and how to attack each section that will make it easier to understand the information. And so the question is like, how do you read a research paper? And not necessarily in the order that they appear. So, usually I for the students, like, what's the first thing you read when you're assigned a scientific paper? And usually you'll get a variety of different answers for this question. Some people will say the abstract, some people will actually just flip to the figures. some people will just go to the discussion and read the last paragraph. So very few will say, I just sat there and read it from the beginning all the way to the end. And so, we all have our own strategies. So, for preparing this lecture, Drew Dennis put a nice article on the web about how to read a research paper. This is how he would attack the paper. He'd skim the abstract, then he would go all the way to the end and read the conclusions, then flip through the results, then the methods, and then go with the next paper. For me, I actually read the abstract, then I read the first and last paragraph of the introduction, because basically the first paragraph gives you all you need to know about what's going on in the field at that moment when the paper was published, and the last paragraph of the introduction actually sums up the whole paper. And then I just flip through the figures. If the data itself isn't rigorous enough, then it's probably not worth my time to go read through all of the nitty-gritties of the paper. And sometimes the figures kind of gives you just a really quick snapshot of the different techniques and the primary data of the paper. And then I'll skim through the first and last paragraph, the conclusion. The first paragraph, usually the first sentence sums up everything they found in that paper, and the last paragraph will give you context about where they're going to use this data and move it forward in the field. So how many times do you need to go through reading a science paper to actually understand it? Once, twice, 3 times, countless, with notes? The answer is yes, and it just depends why you're reading that paper. For example, if you were writing a grant or something and you wanted to, or writing this paper, for example, and you wanted to make sure that you were the first clinical trial ever to study exercise in the context of active surveillance, you have to make sure that statement's true. So you would want to skim through the papers really quickly and make sure this is the first clinical trial. So you wouldn't need to read every single detail to figure out that information. And then the other thing that's really important when going through research papers is that to be very critical of the data that's being presented, because it's always skewed in the view of the authors and may not necessarily be correct. But to read critically, it sounds, like good advice, but it's really hard to do, because to be critical, you really have to be very comfortable and well educated about the problem that they're studying, and you have to understand exactly what they're doing and the approaches that they're doing, and then you have to understand the field well enough to say the approaches they use were appropriate for their problem versus all of these other techniques out there. So to read critically, you really do have to be an expert, which is why peer review is so important to allow these papers to be published in the first place, because right now, in the field, these projects are so big and so collaborative that you really need multiple specialties to even review a paper. And so again, when you're looking at the research, just make sure that it's peer reviewed, and then also in a good journal, and be well informed, and just make sure you just don't believe everything that the authors are telling you. So yeah, so we'll just kind of go through this with our case study, and I'll keep an eye on the time as I'm going through. We've already talked about the title. It's not supposed to be creative or catchy or cute. It's supposed to be informative, and again, also a way to make sure that if someone's typing in these keywords in a Google search, that the appropriate paper will come up. And so this is a great example of a very informed and specific title that lets you know exactly what we're going to hear about in this report. Also, the order of the authors here can tell you a lot about who did the work in the paper. In the medical and basic science fields, typically it's the last author that's the one who's the principal investigator, the boss, who has managed and supported the research to begin with. And so they are also usually designated as the corresponding author. And usually it's the first author who's done probably most of the work. It's probably a postdoc or a student, or perhaps two principal investigators from two different labs have done equal work, and so sometimes they'll put themselves as the bookends of the paper. And then as you move either from first to second to third author, or from last to second to last, that also tells you the importance of the authors and how much work and contribution they've done. So basically the middle author typically is the one who may not have contributed as significantly as those closer to the first or the last. So even just not even knowing who they are, even the order can give you a lot of information. So let's just start with this abstract. It can usually be pretty dense, and this is because a lot of journals, they do have a number of words that length that an abstract can be, and so the authors are trying to put a lot of information in a small space because they have to go through the background, discuss their study, talk a little bit about the results and the conclusions. So sometimes the abstract can be pretty hard to get through, even if it's just a paragraph long. But important because this kind of determines whether we're going to start to commit and read the entire article or not. So for our case study, I just put the abstract here for you. For this journal and for these types of clinical articles, the abstract itself is a little more formatted. You can see it's already being broken down in different subcategories, which actually is makes it very easy to get through this information. But typically, when you're scanning the abstract, you're just asking some fundamental problems. Basically, what is this? What is the point of this study? What main problem are they addressing? Why do we care? Why is it important? How are they actually going to do the study? And what did they find? So, for example, here, men with prostate cancer who are undergoing active surveillance are at an increased risk of cardiovascular death and disease progression, and exercise is shown to improve cardiorespiratory fitness. So this is the problem, and then exercise is what they're going to test. So this is what will be addressed in improving this problem. So that would be, kind of move my cursor here. So that would be the what. So active surveillance patients are highly at risk for cardiovascular disease and disease progression. And then it talks about the effects of disease to address both fitness, cardiovascular fitness, and biochemical progression. And then it'll talk about the design of the study. So for the how, you get a lot of information from just the abstract. So it's already telling you this is a single center, meaning that it's at the University of Alberta. It's a two-group phase 2 randomized clinical study. So the two groups will be patients with active surveillance who have undergone a high-intensity interval training exercise regimen, and those that are not going through extensive exercise. So they're the control non-treatment group. And then, what is involved? It's 12 weeks. They're working out 3 times a week, and the assay will be peak oxygen consumption. But even in this, just a few sentences, I mean, what's hard with the abstract is there's just a lot of jargon and a lot of abbreviations, which makes it very hard to get through some of these research papers. So typically when I'm going through a paper, I'll start jotting down these abbreviations. So HIT, erase, they've already hit us with a couple abbreviations that if you don't catch it fast enough, it'll make it very hard when you're kind of skimming through the paper. Other unclear jargon potentially would be, it's a phase 2 trial, peak oxygen consumption. So some of this unclear jargon, I'll kind of write it down on the side and either hope that they're going to define it in the introduction section, and if not, just do a quick Google search while I'm reading to make sure that I'm not missing any points. And so, for example, a phase 2 trial that they're talking about is typically 20 to 50 participants. And not only are they studying safety, but they're also studying the efficacy of whatever treatment we're talking about. So phase 1, they're really just giving the treatment to the patient and just looking at safety. So it's quite small, and it's not until 3 and 4 where you have significantly larger clinical patient cohorts, both looking for safety and finally benefit of the treatment to the disease that you care about. And then if you're an athlete or kinesiologist, then you would have already known what pink oxygen consumption means, but I just kind of jotted down maximum liters of oxygen consumed in 1 minute per body weight in kilograms, but that's their output. And I talk a little bit about that even in this abstract. This is a particularly detailed abstract, but you can quickly scan and see some methods that are going to come up that we're talking about that are going to measure the oxygen, peak oxygen consumption, and then the biochemical factors that they'll follow would be PSA, some of the PSA kinetics, doubling time, and velocity, as well as in a petri dish, but they're going to measure human prostate cancer growth. And then, scientists don't like surprises, so they're already going to do kind of a spoiler. So they're going to tell you exactly what they found and what they conclude. And so with this paper, what's so exciting is that this exercise regimen did seem to show increased oxygen consumption, decreased PSA, and some PSA kinetics, and decreased cellular growth in their assays. And so, they talk about the limitations as well, and that is that this trial just wasn't large enough, and so obviously more studies will need to be done, but that this was a great start. And then I'm not going to go through this, but I'm just going to give you a snapshot of a typical abstract in a basic science journal. So this is just a paper that I published a few years ago, and the abstract was quite small, but it's more just written as a paragraph versus this type of clinical journal where you're seeing these subsections. But again, I like these because it's very easy to get through and easy to read. Another thing that I love that a lot of journals have started doing now is the visual abstract, which I think is extremely helpful. And so it's kind of like a mini postcard and gets to the point of the design or results and conclusion. So for this particular study, then it's kind of nice to see that they started out with 52 men and then broke them up randomly. Of 46 of them, they broke up randomly into their test group, the exercise group versus the usual care group. They talk about their primary outcome, change in cardiorespiratory fitness from baseline, and it was a 12-week study. And then they even show pictorially their findings. What I found particularly interesting with this snapshot is this particular way of looking at their data isn't seen in the actual paper itself, just in this little abstract, but we'll get back to this in a moment. And then another thing that I like seeing, and it's popping up more and more in different journals, and it's just, again, another tool just to help the audience to get through the paper very quickly if you want to scan it and understand just the key points, both visually as well as in this small little box to the side of the main paper. For this one, which was particularly nice, is that they highlighted the hypothesis of their study. So does high intensity interval training program improve cardiorespiratory fitness and delay biochemical progression of prostate cancer in patients who are undergoing active surveillance? And then they go through the findings and then the meaning of their findings. So their findings would be that it seemed to improve oxygen consumption and decrease PSA and cell growth, and indicates that exercise may be an effective intervention. So everything you need to know is in this nice little box. And journals are going more and more to these nice tools to really help us quickly go through this data. And then, going through these papers, the challenge is really to understand and to identify the big question. So, like what's the main point of this study? For a clinical trial, the big question is usually pretty easy to find, and that would be, does the exercise impact both cardiorespiratory fitness and progression of the disease? And obviously, why should I care? Is this worth my time to read? And obviously, for this particular study, I mean, if this is true, then this is amazing because it's extremely affordable way to manage care. And so we're just talking about the abstract, but we already basically got all of the information and all of the meat from the paper. But again, this particular journal, the abstract was extremely detailed. Sometimes you'll get a lot of that information if it's not in the abstract in the introduction. And the bonus of the introduction for people that are reading outside of their field or are not an expert of that particular study is that you'll get some background information, not only about maybe this particular field, but how the authors think that their study is fitting in the context of the larger problem. So for example, prostate cancer treatment options. And then it'll also, you know, again, there's just a certain template that these science papers will follow, but the structure is usually, it'll describe the gap in knowledge, the rationale, you know, what research came before this paper in order to make this a valid and worthwhile study, the specific hypothesis that's going to be tested in this study, and then the conclusions. Because again, even though we're in the introduction, they're already telling you what's happened at the end of the paper. And then for me, if you're scanning papers or you just don't want to get into the minutia, but you want to understand the big point of the paper, I usually skip to the last paragraph of the introduction, because this is usually where the authors most clearly will tell you the hypothesis to be studied, how they're going to do it, and what they found. And they'll do all of that in about 3 sentences, which is really nice. And so for our case study, I have highlighted the introduction, which is actually not too long, which is nice. And then just kind of highlighted where these pieces of information that you will typically see in every scientific journal article reside. So the problem, so they're discussing how men with prostate cancer on active surveillance have approximately 3 times higher risk of cardiovascular disease related deaths than prostate cancer deaths, and 30% will progress and require treatment within 3 years, or 55% in 10 years. So that's the huge problem. And then the question would be, you know, can exercise boost both cardiovascular disease health and delay disease progression? And so they also kind of picked out the rationale that there's already research out there before this paper was even published showing that exercise improves cardiovascular or cardiorespiratory fitness, that aerobic exercise has been shown to suppress progression of prostate tumors and metastasis. So they're giving you these little references of papers that have already been published before this that they're building their work on. And then the gap in knowledge, they're telling you that only 1 clinical trial so far has ever been published that examined exercise in the context of active surveillance, but they never investigated efficacy, you know, whether or not they saw any difference. And then they clearly discussed their hypothesis. So we hypothesized that high intensity interval training will generate substantial improvements both to health-related fitness and biochemical progression of prostate cancer in men on active surveillance. So again, you know, even if you didn't get through the whole paper, you already are hitting some key points just with the abstract and the introduction, which is useful. But again, it doesn't matter what field you're in, there's always just a lot of scientific jargon that's really hard to understand. And so, you know, people have different strategies. You can skip the word, write it down in the margins and figure it out later. I usually Google while I'm reading just to like pop up certain terms, or maybe there's some abbreviation that's driving me crazy, or a little term. So yeah, so I'm always either on my phone and Googling while I'm reading a paper. You know, you can skim the paper and hope that the author defined it. Sometimes they do, sometimes they don't. You know, ask a colleague, grab notes. There's all different strategies, but you know, you're going to run into some of this scientific jargon. There's no way we can be an expert on everything, and so you know, there's just different strategies. So again, you know, diving deeper, Google keywords, look up references cited in the paper itself. If you think it's a pivotal paper, but you're not comfortable of what's going on in this field, picking up a review that, you know, someone has synthesized, you know, like years of research for this topic, that's the fastest way to catch up. You know, basic scientists do that all the time because we're always moving from one field to another. Sometimes our factor, you know, maybe you think that you're studying cell death and suddenly your factor is really important in metabolism. So we have to like quickly catch up in these fields. Another helpful thing is to look up commentary. If the paper is really splashy or it's really gained traction, both in the scientific media field or in the general public, then some commentaries will be really helpful. They're kind of like pre-digested, so they've kind of read the paper and then they've kind of restated it a little clearer for the general public. So, you know, I just did that. I just kind of Googled the title of this paper that we're talking about that really just came out like 2 weeks ago, and there's a lot of media and talk about this paper already. So if you go through here, you know, they've already kind of pre-digested the paper and kind of put out the key points, and maybe in a different context than how it was written in the paper. And again, also if they're talking about a rationale and they've kind of highlighted, you know, like exercise was already found to decrease prostate cell growth, and you're like, I wonder who did that. You know, maybe that's a really interesting study. They've referenced where they've got that information, and the references are in the back of your research paper. So this can also point you to other pivotal papers that you might want to read, which is a nice tool to have. And then in the paper, I mean, it has to be reproducible so that other people can try and see if this works in their own labs or in their own settings. And so there's always a material and methods section, unless you're going to try to, you know, start up a new experiment in your lab. Sometimes you don't really need to linger too much in the weeds if you just want to understand what therapies are out there. Or sometimes when you're reading the results, you would just kind of flip back to certain methods just to make sure that kind of clarifies how an experiment was done or how the clinical study was set up. So, you know, you can kind of skim through here and then go back when needed. In this paper, the first figure is actually referred to in the methods section, and this was just talking about their patient recruitment, and that they actually started out with 361 men, 176 were eligible for their study, and then of those, those that actually consented and wanted to participate, they were all the way down to 52, so 26 and 26 were randomly put in two different categories. One would have the high intensity exercise and the other did not. So that was a nice, useful chart for me. And then the other part of the methods section, you know, there was a lot of stuff in there, but you don't have to read it all. But the one thing that was interesting was, you know, there's this exercise program that they call HIIT, but what actually is it? High intensity interval training. And so, you know, it was interesting to read about that. So it was an exercise session, 12 weeks long. They worked out 3 days each week. It was on a treadmill. They had a 5-minute warm up. And so, which they defined as 60% peak oxygen consumption. And then they had more intensive training for 2 minutes. So this was basically at max oxygen consumption. So they're really working out intensely. You know, they're part of this calculation is that they're, get high gas exchange, that the heart rate is up. And then they have 5 minutes of cool down, and they do it's interval training. So they do this whole cycle 5 to 8 times for each session. And then as the men are moving through their exercise program, their stamina is increasing. So they actually extend the exercise time as well. And then it'll also tell you, what about the other people who aren't exercising? Well, they just had normal exercise levels. But also what was nice for these men, because this clinical trial did show a positive outcome for the HIT program, is that they offered these men a 4-week HIT program at the center, or a 12-week community-based exercise program. So I thought that was interesting. And then for the results section, and we can just go through this a little quickly, but obviously this is all the meat of the paper. So you actually see the data, the figures and tables are within the results section. One caution though is just don't believe everything you read. I mean, when we're training PhD and master's students or medical students to read research papers, it's how to read things critically. Usually we're always kind of like, don't believe it until you believe it. I guess for pessimists, I'm not sure, but be critical when looking at all the data or the experimental setup. Ask questions. Subheadings, sometimes the results are broken up in subheadings. This is really helpful if you're just skimming and trying to figure out, you just want to get the gist of the paper. And then for me, if I want to get a quick look, like maybe I'm trying to figure out if I'm reviewing a manuscript and I just want to see how rigorous the study is, like I just flip through the figures first. For me, if they're using the same technique, over and over again, and it's not very rigorous of a study. So for our case study, table 1 just described the patient clinical data, which was nice. I mean, they gave us a ton of information for a clinical trial. You're kind of like, why do I need to know all this? But it is very important. I mean, it doesn't address potential unintended factors that could have skewed their data. So, obviously prostate cancer is a great example of race disparity and outcome for the disease. So you might want to know the race breakdown of this study, the age. They're telling you about education, their medical history. Did they have other health issues going on besides prostate cancer? Then for me, I was kind of looking at their clinical stage breakdown, and I was wondering, where you guys had a discussion, it sounds like, about Gleason score. Gleason score 7s are always a hot issue for me. So I was wondering why they did have a T2B and a few of these 3+4 patients in here when the majority were Gleason 6, because that could skew the data. So again, for me, I'm just always kind of thinking kind of critically and just looking at potential issues with the data. And then they also talked about the exercise behavior. And so here's some standard deviations. Obviously none of the men before the study were vigorous exercisers. And so they were just kind of breaking this down, but there must be a huge range of exercise for these standard deviations to be so high. And when I say standard deviation, it just means just a measure of how exactly what this says, how dispersed the data is in relation to the mean. So if you're doing a study and say, if you're studying cellular growth, and so they all grow, 50% would be your number, the result. If you're getting data and it's like, it'll grow 10% faster or 100% faster, I mean, there's a huge spread of the data. And so you're not quite sure if your data is all scattered, whether your result is due to the treatment itself or it's just kind of random scatter. So the closer all of your data points are to the mean, will have the lowest standard deviation, and that's what you want to see for a really good, rigorous, and reproducible result. So we keep an eye on standard deviations. So for table 2, this is just, not this is just, but this is the research for their study. So this is their output for their 2 clinical groups. So the exercise group, the HIT group, or the control group, and they're looking at all sorts of things. I mean, obviously we care about their data for cardiopulmonary fitness, but they're also telling you some biochemical outcomes. So they're measuring PSA levels in the serum, doubling time, and velocity of the PSA, so some of the kinetics as well. They're measuring testosterone levels, and then they have this interesting in the Petri dish assay as well. And so when you're looking at all of these numbers, you're also asking right here, are all these data points and results, are they statistically significant? And so in this last row here, it says p-values for adjusted between group differences. Your p-value should be less than 0.5 to be statistically, sorry, 0.05 to be statistically significant. So yeah, so these, let me just move my window here. So these numbers are a bit high. Sorry for the typo here. So 0.05, sorry. There we go. And so, oh, I have a typo here too. So 0.05. So what is a p-value? So for the p-values, it's the p-value is telling you if your hypothesis is that exercise will increase your cardiovascular fitness, then the null hypothesis would be that the exercise did nothing. And so for your p-value, it's actually scoring the null hypothesis, which would be basically exercise does nothing, 0. And so within this distribution, or the area under the curve here, if your treatment is doing nothing to your study, then your data will be staying very close. About 95% of your data will be under this curve here. But if your treatment is actually doing something significantly, then you'll be out here in these very unlikely observations, meaning that if something truly was happening that was significant, then it would be very far away from this null hypothesis, basically. So your p-value should be less than 0.05 to be statistically significant. And so looking over here then at their cardiopulmonary fitness or their PSA values, so 0.01, 0.03, 0.04, these are all statistically significant values. 0.1, 0.24, these are all very high. I'm sorry for the typo here. And these are all higher than the 0.05 cutoff for a p-value. So what this is telling you is that their treatment regimen was having no impact on these particular data points, which were doubling time for PSA, as well as regulating testosterone levels. So, when you're looking at the data, just kind of stare at these p-values and see if they're significant or not, and this will help you in interpreting some of this data. Also, they're doing something showing you the mean, and they're showing it in relation to 95% confidence intervals. And these are, again, it's kind of a similar idea. So these are ranges of values where you can be 95% confident that it contains the true mean of the population. So in this case, if exercise is doing something significant, and you take the mean of all of your data points, say exercise is increasing proliferation by 50%, that would be, where your mean is falling, and then you're calculating distribution of your data that is still truly significant, should fall on either side of your mean within this 95% area under this curve. So any outliers here, it's basically describing your lower and upper limit of your data that would be considered significant. So any of these outliers here that are outside of these confidence intervals means that it's probably not doing what you think it's doing, basically. So you're just not very confident in those data points. And so that's what it's just defining that spread to the mean. And so if you have data points outside of there, then it's not significant. So looking at some of these values then, and everything in blue, I'm just thinking about going through this paper, though I'm not a clinician, and just asking myself, do I believe in this data based on the values that they're giving me? So, is this data statistically significant? What about the standard deviations? So for cardiopulmonary fitness and their peak oxygen consumption, some of these standard deviations seem quite high to me. So this is saying that peak oxygen consumption for the exercise group at baseline versus after their exercise regimen are 29.6 and 30.4. But this means that they have some patients that had, like 20, whatever, like 24 or, 32. So there's a huge spread. And that spread is also overlapping plus or minus 6. So they have 36, 30 or 24. So there's not a lot of difference, honestly, when you're looking at the data error in these numbers. What's interesting that these authors have done as well is that they're looking at these values, but then they're adjusting between the group differences. So not taking consideration their standard deviation and just taking a mean change and then adjusting for that and then taking their p-values. So they're kind of, for me, I think they're kind of skewing the data to look a little better and a little tighter than the raw data actually is telling you. So I was just a little bit uneasy about how they're doing a mean change and adjusted group differences. And then also, as I alluded to before in the visual abstract, they're showing you a figure here that is not seen in the paper, but they're actually taking data from their cardiopulmonary fitness data here. So they're actually grafting this data out here. But they're not showing you error bars, which would be quite high for both of these values here. This is the baseline before exercise, both for the exercise group and the non-exercise group, but they're quite different. So I was just wondering if this is truly randomized. The baseline should be the same for both groups of patients. And also they're trying to say that there's an increase in cardiovascular fitness before exercise compared from before exercise to with exercise, but these differences are very small. Actually, they look larger because their y-axis scale is quite small. And also with the error bars, I don't think they would be as significant. So again, I'm just kind of walking you through how I think about these types of analyses and how, I don't know, I'm always kind of just questioning the data. And just looking at the unadjusted data for PSA, kinetics, as well as testosterone. So again, for example, for PSA velocity, they've actually showing you the standard deviation curve, like this is their mean, but patients were showing both above and below these values. And so that's where you're getting these error bars. If these error bars are overlapping, then they're not significant. So even though when they do some of their between the group differences to get their p-values, the raw p-values I don't think would be statistically significant. So I'm just kind of highlighting, read critically, kind of question the data, and just don't always believe it, you know, on face value what the authors say, because there may be some over-interpreting for these studies.
Yeah, Aurora, I think we
Yeah.
got to leave some time for some questions.
Oh, sorry. Yeah, sorry. Yeah, so I'll just wrap up if that's okay. I'm sorry, everybody.
Sure.
You could just kind of scream at me. But anyway, so yeah, so I guess just kind of taking a peek in my mind how I would go through some of this data and just to read critically, and you know, I just kind of was just over dissecting some of this data here and how they were interpreting it. So, but then again, what's nice is that you can get through the nitty gritty and overanalyze. But if you just want just to get the overall gist of the paper, another opportunity would be the end part in discussion. And again, they'll sum up all of the major findings, which are nice. And what's also nice is if it's a good paper, they'll give you a well-balanced take on their own work and give you both strengths and limitations. So for this work, the strengths was the novelty of the study, but the limitations were that it was a small group and that they just need follow-up, which is, I think, when I'm kind of nitpicking with p-values and a lot of error, it just means they just need larger numbers to kind of clean up some of this data and see if there really is a significant change with exercise. So yeah, so I guess that's sort of my take home to, you know, find the right paper, to make sure you want to invest the time in that paper. And then when you go through it, to just read critically, take notes, you know, talk to others and see what their take is, because from different perspectives, you'll get, you know, different evaluations and different thoughts about the work. So yeah, so hopefully that'll be helpful. I mean, these last slides, I was just kind of summing up where the field is, and you know, there's a lot of breakthroughs in the basic science arena, thinking about, you know, different alterations in prostate cancer, and this will lead to different markers and screening tools and therapeutic targets. And the most big late-breaking FDA approval was in December for this PSMA targeting and PET imaging, which looks great. And also as a therapeutic, it's looking great. And we're also using PSMA targeting in our own lab for some of our factors. So I was super excited. But yeah, so just the take home message, you know, skim the paper. You know, there's a template, so just don't get too bogged down with the vocabulary. You can Google while you read. You know, it's nice, it's already broken down section to section. And then the most important part, but the hardest part, is just to reflect on how, if this is impacting how you think about the field or treatment options, and just to be critical. So that was it. I'm sorry that I was kind of lingering.
Oh, no reason to apologize, but thank you for your presentation.
Sure.
And our founder, Rick Davis, says he's years away from his statistics courses, and he asks, how can I get a quick and easy refresher course on p-factor, well, p-values and so on? Well, first of all, everybody should realize that it's a different kind of p than this group usually is worried about. So but where can we get kind of a quick and easy review on statistics?
Yeah, I think that's a great question. You know, there's a lot of resources, honestly, on YouTube, And so there are a lot of institutions. MIT has a lot of nice, like quite short videos about all sorts of things. So really, if you just type in p-value, you'll get some really nice and short video explanations. So I think that's honestly a great resource.
Are there any books out there? I'm not sure anybody reads books anymore.
I know. I mean, in my lab, I do have, I should probably run over and get it. I just have a couple just super basic statistic textbooks. For our students, I mean, they all take statistics. For me, I just collaborate with biostatisticians. But if you just want basic statistical explanations, honestly, YouTube has some great resources out there. But if you want to do more focused ones, you might want to look in the YouTube arena for MIT has some nice resources. Cold Spring Harbor as well.
I have a question. If you follow, the reports in the daily media, press or video or TV or whatever, for example, one week they'll tell you that coffee is the best thing ever and it's going to help you, with your cancer or whatever. Then the next week there's a contradictory study. How are consumers, to interpret this? It's like for every MD or PhD, there's an equal and opposite MD and PhD. I mean, how you say to read these things with a critical eye, but how do we do that? Maybe Herb Geller, who's a researcher on our board, would want to weigh in here too.
Let me see. Am I muted, Howard?
I
No,
can hear you.
No, I'm not. Good. No, I think there's, first of all, I think this presentation was really interesting in terms of the paper, Aurora. I personally, so I will tell you, I have a little bias in statistics because my wife is a biostatistician and was the president of the American Statistical Association. So, and she does clinical trials. So I have a little bias there. And I think, the answer is that science is not static, right? As new data come in, and then we've seen this with COVID, right? As new data come in, you have to change your hypothesis, you have to change your view of what the world is like. And I think that's one of the joys of being a scientist, that it's never done. Now, I think that the issue for me, though, is how do we determine whether, number one, whether a paper is really significant, and not just that the p-values are significant. And I think this is where I think it takes a lot of effort.
Yeah.
So
No, I agree. And I think that's a hard question, right? I mean, how someone without a lot of expertise would be able to critically read the paper. I think that's why it's also nice to, read reviews and talk to, what you're doing right now, inviting clinicians or basic scientists, statisticians to come in and weigh in as well. It's hard.
Years ago, I would go into medical libraries at Northwestern or University of Illinois in Chicago, and I would have access there to the journals and so on. And I wonder these days, especially in this time of COVID, how can we get access to these journals?
okay, well, Howard, I can answer that question for you. So I think one of the most wonderful advances in scientific literature is there's a movement called open access. And what we're finding is more and more journals are open access, which means they're available to everybody from day one. And I think many of the medical journals, and I think soon everything will be open access. And I think, so if you go and find an article that you think you want to read, chances are you'll be able to get it by downloading a PDF. Now, there's another little secret. I mean, I'm a little older than Aurora, and so she may not remember the days when we used to get reprint requests from people.
Yes.
So if I published the paper, somebody would send me a postcard and say, "Can you send me a copy of that paper?" But I think another way around it is, if you find a paper that you're interested in, send an email to the author and say, "Can you send me a PDF of your paper?" And I would be very surprised if they don't.
Well, in fact, I use something called ResearchGate,
Right.
and
Right.
and you have access to some papers there that can be downloaded, and you can connect to the authors. So that could be one way. I think it's open to anybody.
Yeah, no, I use ResearchGate as well. So I think you just have to create an account, and then you can request the authors. And usually about 70% of the time they will send you the PDF. Or if you're on ResearchGate, a lot of people will just put their PDFs on there, so you can just access it without asking them. Hey, I have a quick question. How do I stop sharing? Because I can't turn it off.
It should be at the bottom of your
Trying.
screen. The bottom of your screen, Aurora.
If you maximize the window, you should see a green
Oh, thank you.
or red at the
Yeah.
top. It says stop share. I wanted to
Thanks,
say
Larry.
something, Herb Howard, that it really was 40, 50 years ago that I was into biochemical research. And that figure, 0.05, p-value 0.05, still is in my head. I don't remember the courses that I took that led to it, or the standard deviations and the statistics. I can't imagine wanting to go back and recreate all of that math. Just to keep in mind that when I look for that figure that Dr. Aurora is talking about, and when you see the high p-values, it means that there's a lot of variation and it's not so predictable. And then the preponderance of evidence. You see a lot of all these talking about, one expert says this and one expert says that, but you start to watch preponderance start to shift, and then you start to form your theory. So some ways it's extraordinarily complex, way above any of our pay grades probably. But in the other, it's sort of just a few facts, and like not jumping at small studies or small conclusions, and keep your eye on the ball and watch when things sort of snowball and build, and say, oh look, this is becoming something serious, especially when it applies to us individually. We like follow the things that matter to us as cancer patients, and not as trying to be the researcher or understand. But Dr. Kersher, I loved your presentation. Really was the skeptical way, inquiry way in which you look at it. So thank you guys so much. And none of us are going to become brilliant statisticians in our senior years.
Yeah, I mean, it's definitely a tricky field. I mean, the good thing with science for us is that we always have collaborators. So I have a bioinformaticist I collaborate with, and a pharmacist, and a biostats person. So you have to know enough to know when you're wrong or when you're right, but you can also talk to the experts. So it is useful for sure.
Aurora, let me ask you another question. You're in a virtual room filled with guys with prostate cancer. As a prostate cancer researcher yourself, do you have any questions for us?
I do. Thank you. I mean, for me, like with that presentation, for me, that was really hard to prepare because I had a lecture prepared geared towards graduate students and medical students, but to try to figure out how you want someone to help you to go through primary research, or how to make us better in conveying what we're doing. I mean, I guess I'm wondering, what did you want to hear actually for this type of presentation? For me, I was trying to put myself in your shoes, and that, for me, recently my husband was diagnosed with esophageal cancer, and so for me, like, it's a rare cancer, and I just didn't know a lot about it. So when I was thinking about reasons to want to read a research paper, what I didn't have on there that I put on there for tonight's talk was to educate and self-advocate, and that's what we had to do when we were learning about the disease, learning newer treatments, and then really self-advocating for different treatment options. So when I was listening to this earlier this evening, saying how doctors don't always want to be asked, what their expertise is, that's exactly how we approached my husband's treatment, because the surgeries, it's only 1% of all cancers are this type. And so it's a numbers game. So when we were talking to surgeons, we were always saying, what are your complication factors? How many operations do you do a year? And things like that. So I guess for this group, how can a basic researcher communicate better to you, is my question.
You made the point of education and self-advocacy, and I think that's the whole point of what it is that the NCAN groups do. So, I gave you some questions, and from a patient point of view, and you answered them. So I don't know what else I would ask you.
Yeah.
So.
Oh, Mason, I can't hear you. Your microphone.
Turn your mic on, Mason.
There. Okay, Rick. Just give me a second. But these limeys, I don't know about. Just hurry. My name is Mason. My question kind of revolves around. Is anyone here at the site familiar with an author whose name is Bill Bryson, or a newsman whose name was Jeff Greenfield? They had an innate talent for taking complex situations and ideas and breaking them down into easy to understand ideas and notions. Bill Bryson once spoke about, if the sun were the size of a pea, then the other planets would be these particular sizes. What I'm trying to get at here is, you early in your presentation mentioned some sites where we could find and identify papers. I was wondering if any of those sites deal specifically with non-professionals. I can only speak for myself, but trying to understand some research papers on top of coping with prostate cancer becomes a bit overwhelming. And I think all of us here, and as Howard mentioned, one of the wonderful things about this group is it is peer-led. We help each other out. We listen to what other men have done that has worked for them. Yeah, that was kind of a question. I'm apologize, I'm being unclear about how to phrase it, but of the sites that you mentioned early in your presentation, do any of them pertain specifically to easier to understand breakdowns of these papers?
Yeah, so a lot of the big hospital websites, and even the Prostate Cancer Foundation, they sometimes have at least email blasts, or even on their web page, they'll kind of break down like what's the latest research, and then they will describe in very clear and brief sentences, like what the latest papers are, and sometimes they'll even have some like interviews with some of the authors of these papers. So I think those types of sites are really useful. So I mean, the Prostate Cancer Foundation does that, like the Mayo Clinic, MD Anderson sites. So I think those would be places that I would start.
I do want to thank you for interjecting your husband's personal story in this, and in a way, it's good for me to hear a well-spoken person like yourself say that you encountered problems in understanding these papers when you first started to read them. Thank you.
Thank you.
Yeah, there's a couple of things I want to just say on this. One is I just want to add MSKCC. If you sign on to some of Memorial Sloan Kettering's newsletters, they do a good job at explaining a lot of the research for a lot of different cancers in easy to understand language. But you can't be picky. What they give you is what you get to read. It's not like what Mason wants to do, where he wants to go in and find something and he wants that in the easy to understand language. We're getting there, but we're not there yet. The second thing I wanted to say, Aurora, is we have a couple of groups for you. And we'd love to invite you. We'd love to attend. I'd love you to attend. We have a caregivers group, a cancer caregivers group, and the issues that you're talking about are frequently addressed by some of the women that and the men that attend that. We've got a woman whose husband is dealing with appendix cancer, and it's a rare cancer, and she's facing the sort of issues that you're facing. And so you are very, very welcome to attend that caregivers group. I think the next meeting is the first Tuesday of it'll be the 7th of next week, and come on in. And we have a group for your husband.
Thank you.
We have a group for your husband too. We have a Speaking Freely group, which is to talk about all the issues around being diagnosed with cancer except treatment. And as you well know, and this question came up earlier on today in a meeting that I was in, a lot of times men don't do a great job dealing with the mental health and the emotional and the anxiety side of cancer. And this group is to allow them to do it. And that meets on the third Thursday, I think the third Thursday of the month. And these are all free and drop-in. So you just have to go to our website and find them.
Great.
And that's a men-only group, I'm afraid. We actually had to kick a lady, a caregiver, out of that last meeting. But that's a men-only group, because a lot of these issues that these men want to talk about, they're not so comfortable talking about it in female company. So, but we'd love to see you in those groups. That's why we run them.
Yeah, that's great. Those are great resources. Thank you. Well, thank you so much for letting me present. But yeah, definitely if you have any thoughts or things or questions, or you're looking for a paper, just drop me a line. I'll be happy to help.
Well, thank you. I see. I should put my screen on because I see that Howard dropped out.
Right.
And
Well, yeah, my internet went out.
oh, okay. All right. Well, you're back just in time, Howard. Although I was just about to grab your hat and turn on my camera, but I don't have to. I like that hat. You know. Go ahead. Go ahead. I was going to let Herb in, and then it's all over. Okay.
Well, I want to thank everybody who came, especially want to thank Aurora for sharing with us everything that she did, and that's all that we got. Sign up for our webinar with Dr. Klotz, September 29th, and that's all I got. Anybody else?
No, but I do want to
I'll
say
try
one
to keep this short, Rick. I was just wondering if Aurora, you would, if you happen to come up with any ideas or see articles revolving or dealing with this subject, please contact one of us. We'd love to, keep you within the circle here and be able to draw on your expertise. We appreciate it.
Yeah, absolutely. Yeah, it's been a pleasure. Thank you so much.
And Aurora, I do want to say that that praise from Larry Fish was extraordinary, because it's not often that people hear such accolades from Mr. Fish. So that was truly something special.
And it's all recorded too.
It is recorded. So he cannot be denying that he was praising you ever again. That sort of praise. We don't hear from that. I almost fell off my chair.
For what it's worth, I'm not sure, but I certainly stick with what I say.
Good. Thank you.
Just the pluses and minuses, just the way you approach the research. And I said skepticism, but I meant with, careful consideration for what things really meant, and considering the possible biases and everything, the back and forth of the way your mind worked. It was just wonderful to hear that.
Okay, thanks.
Okay.
Great. Well, all right. Thank you so much.
You're going to be definitely welcome at some future sessions with us.
Awesome. Yeah, please invite me back. I'll fine tune my presentation if I ever need to get it again.
Thanks a lot. Okay, Joe, right. You're in charge. You got the off switch.
Just before we hit the off button, back in the chat again, I put the information for the upcoming the webinars with Klotz. So please go take a look at it.
And I should add, Howard, that at the end of the month we've got another great webinar with Nancy Novak. I don't know if any of you know Nancy Novak or Nancy's List. She's been gathering this list for probably 10 years or more. It's a wonderful website with all kinds of fantastic links, mostly for cancer resources. And she's going to present her 10 favorite links. She's going to talk about her 10 favorite cancer links, cancer resource links, and that's going to be the day after Dr. Klotz. So, just saying,
We'll make sure that we have that in the reminders, and we can make sure that we post that, in our meeting next week as well.
Yeah, we will. But if you're interested ahead of time, just look up nancyslist.org. Okay.
Well, call it quits. Good night. Thanks, everybody.
Good night. Thanks, everybody. Good night.
Good night.
Bye-bye.